It's time for a new unit, that new unit being cell growth and reproduction. Our initial topic for this particular unit will be on spontaneous generation. In terms of spontaneous generation, there are 4 names that are of some significance, and you can see those listed here to go along with an assignment that you're going to be doing. Francesco Redi, John Needham, Lazzaro Spallanzani, and finally Louis Pasteur. Redi attempted to end spontaneous generation by showing that maggots generated from eggs laid by flies and not being spontaneously created from rotting meat. John Needham, on the other hand, was a supporter of spontaneous generation. He believed that gravy, despite being heated, would produce microscopic organisms generated from the gravy itself. Lazzaro Spallanzani, on the other hand, didn't think much of Needham's experiment, decided that he had not heated the gravy long enough or hot enough to truly kill everything that was inside, and so he redid Needham's experiment. In the end, though, it was Louis Pasteur that finally put an end to the idea of spontaneous generation with his famous swan-necked flasks. What is spontaneous generation exactly? The belief that living organisms can form from non-living material. And as was just stated, spontaneous generation, or abiogenesis as it is also known, is false, proven so by Louis Pasteur. What is true is biogenesis, the belief that living organisms are formed from other living organisms. Given that that is the topic, what that leads us to is reproduction. Reproduction can come in 2 different basic forms, sexual and asexual. With sexual reproduction, the key is that there are 2 parents. And because there are 2 parents, they are going to blend together their genetic material to form a child that is unique, different separately from both of those parents. Asexual reproduction, on the other hand, is different from sexual in that there is only one parent. And because there is only one parent, that means that the offspring must be genetically identical to that parent. One form of asexual reproduction that is practiced by prokaryotic cells would be binary fission. As you can see in the diagram to the right, we have a cell at the top, a prokaryotic cell, an organism unto itself, and it is filled with its massive clump of DNA. For binary fission to occur, all that has to happen is that all of that DNA needs to make an exact copy of itself. So imagine that this lump over here on the left side is the original DNA that was up here, and then the lump over to the right is an exact replica of what's on the left. Once that has occurred, and as you can see it has down in our third picture here, all this prokaryotic cell needs to do is to simply split itself down the middle, and you'll end up with 2 cells where you once had one, and they should be identical to each other. Asexual reproduction in the form of binary fission. To continue along with that same idea of reproduction, some sort of tangential topics, one of which being conjugation. In conjugation, we again have bacteria. You can see 2 bacteria here to the left. And you can see on the one on the left, there are these hair-like structures sticking out, and those are called pili. Pili are typically used by bacteria to attach themselves to surfaces. But some of those bacteria have pili that are special. They have specialized and are referred to as sexual pili. And that's what you see connecting the 2 different bacterial cells here. If we look at this version of events that's trying to represent the same thing, you can see the bacteria on the left, you can see the sexual pili, and it's going to connect that to the bacteria here on the right. That pili is nothing more than an open tube. How does this come to have any connection to reproduction? Well, bacterial cells have their main DNA ring, but sometimes have little snippets of DNA called plasmids. And of course they contain genes, and genes code for particular proteins. What can happen is that when a sexual pili, an open tube, connects these two bacteria, it's possible for that snippet of bacteria, that plasmid, to be transferred from one bacteria to the next, thus passing on certain traits. Now, is this reproduction? Not exactly. But if you think about reproduction in the sense that it really is just the passing along of genes from one individual to another, then this kind of qualifies. In a typical version of reproduction, we are passing our genes down vertically, in other words, from one generation to the next. This example of conjugation is really horizontal. We have here two members of the same generation, but they are transferring genes from one individual to the next. One last reproductive strategy that we can mention when it comes to bacteria would be the formation of something called an endospore. Now you're looking at a prokaryotic cell here, and yet it does not look like a prokaryotic cell. It looks like it has a nucleus. But we know that that cannot be because prokaryotic cells don't have a nucleus. Instead, some bacteria have a reproductive strategy that they use in which they form an endospore. Endo simply means inside. And so you're looking at a spore, or a protective coating inside the cell surrounding the genetic material of this bacterial cell. Why is that a reproductive strategy? Well, because what these particular bacteria can do is to essentially go into hibernation. If the environment around this bacterial cell is poor, why would that bacterial cell want to reproduce? Why make more when the environment really is trouble in terms of supporting life? So instead, an endospore can form inside this bacteria. It is therefore going to protect its all-important genetic material. And as I said earlier, the bacteria can just basically go into hibernation. A year, 10 years, 100 years, and it can basically just wait out the circumstance. And once the environment changes, maybe it's the temperature that needs to be better, maybe it's the amount of moisture or the available nutrients. Once those circumstances are better for the bacteria, it will then break down this endospore and immediately start to go into reproductive mode, asexually making more copies of itself.